So firstly, I would like to thank Lei and Kostya for their nice introduction. And I'm very happy to be invited here to have a presentation. Mm, and what I'm going to talk about is more focused on a specific topic. So it's about algorithmic development to study cell communication using single cell data and spatial transcriptomic data. Uh, so I would like to introduce a bit background first. So as we all know, cell communication is restricted by cell location. There are four types of cell communication. The first one is autocrine signaling. It's a way a cell communicates with itself. And a cell can also communicate with its direct neighbors through direct signaling. Also, there is paracrine signaling. A cell can communicate with its nearby cells through ligand receptor interaction. And the last type of communication is endocrine signaling. It's where a cell that communicates other cells from a distant organ by transporting the ligand through blood vessel. So as we can see here, cell location is very important in cell communication. However, this information is lost during single-cell RNA sequencing. Here comes spatial transcriptomics. Spatial transcriptomics is a technology that captures both the transcript information and spatial location of part of the cell, single cell or multiple cells. Based on the resolution of the technology, there are four types of ST sequencing. And the first one is sub-cell resolution ST sequencing, such as 6-scope, pixel-seq, and stereo-seq. And there are also technologies that can detect the location and transcriptome and single cell resolution, like segfish, merfish, and StarMap. And there are also near cell resolution ST technology, which detects the transcriptome from a region whose size is roughly equal to the size of a cell. That's why we call it near cell. And the representative for technologies for this type have SLIDE-seq and SLIDE-seq V2. And the last one is multi-cell resolution ST technology. It mainly includes VISM, and it's also a commercialized method that has been applied on many studies. And here is the schematic for the barcode based on ST technology. So basically, it's a barcode that contains the spatial information was added on the end of the RNA molecule, and through this barcode we can know the location of the RNA molecule. And this is the schematic from SLIDE-seq V2. And here is a table that compares the specifics of different technologies. So for barcode based on methods like VISM, SLIDE-seq V2, 6-scope, stereo-seq, and pixel-seq, they can detect RNA molecules unbiasedly, but some of them lack sensitivity, like VISM, the sensitivity is relatively low. And the sensitivity of the technology has been improved since SLIDE-seq V2, but unfortunately it sacrificed the resolution of the sequencing. It can only detect at near cell resolution, and more methods that can detect the transcriptome and subcell resolution have been developed, and these methods improved the sensitivity significantly. But however, these methods are still very new and it's not easily accessible. So we don't have much data for those subcellular sequencing technologies. And there are also single cell resolution ST techniques like segfish, merfish. Those methods are based on image based. And the shortcoming of these methods are that they couldn't detect the transcriptomes unbiasedly. They can only detect part of the genes, like at most 10,000. But normally you cannot detect so many genes because it's very expensive and time consuming. And these methods also have like much better sensitivity with them, but however, they don't have enough throughput to study the whole transcriptome. So as we can see, current spatial transcriptomics still lacks throughput, sensitivity, or resolution. And just to summary, so we already know cell location is very important in cell communication, but single-cell-seq lost the information during sequencing for single cells. And current ST sequencing still lacks throughput, sensitivity, or resolution. So we need a tool to bridge the gap between the spatial transcriptomics and single-cell-seq sequencing to overcome the disadvantages. And that's why here comes spatial inference. So it's basically a strategy to map single cells to ST voxels to restore the location of single cells. In this way, we can transform the low resolution or near cellular resolution ST data into single cell resolution. And this idea was first presented by Akim and Sura in 2015. Afterwards, then more tools were developed, and the newest three methods are LovaSpark, SPA, OTS, and Tagrim. And here are some brief description. Here is some brief description for the three tools. So there are many based on distance or the similarity between single-cell RNA-seq data and ST datasets. These methods can work very well on small datasets like Segfish or Vism, using only small number of genes to run their algorithm. But when it comes to a large dataset like statistic V2, near cellular resolution ST data, they don't work that well as how they work on small datasets. And that's why we need a new tool that can be applied on large dataset from near cellular resolution ST data or even higher resolution ST data. And that's why we developed SZ-SPA. So it's a tool to map single cells to spatial coordinates. The model is the core component of the model is a deep neural network. It's firstly trained on ST data to learn the relationship between the gene expression and the spatial coordinates for voxels. After training, the model can learn how the gene expression influence the location of single cell, a cell or a voxel, and then we can use the trained model to predict the location of the single cells. And here are two examples that show our prediction. We benchmark SZ-SPA against other tools and one simple logistic regression method on three different datasets using three metrics: Gerson's rank correlation, root mean squared error, and R squared. The red bar shows the performance of SZ-SPA, and the other colors show the performance of other tools. We can see SZ-SPA outperformed other tools on all of the datasets according to the three metrics. And here we can show like more intuitive images to see how SZ-SPA outperformed other tools. So the first figure is the true location of ST voxels, and the second figure showed the prediction from SZ-SPA. And we can see SZ-SPA recover the location of the voxels very much better than other tools. And that was so. This is for the statistic V2 mouse hippocampus data, and we also benchmarked mouse embryo data from statistic V2. So the first is the true location of ST voxels, and the second one is the prediction of SZ-SPA. We can see the other tools just couldn't. Other tools just couldn't recover the locations, exact locations of the voxels, while SZ-SPA recovered the locations very well. And some of you might be wondering like why SZ-SPA can be outperformed significantly so much than other tools. And here we can explain in this dataset. So in this dataset, we benchmarked these tools on Drosophila embryo data. We can see here SZ-SPA didn't outperform, have so much like advantages over other tools. That's because like here the number of cells involved in the dataset is very limited. Only maybe thousands of cells are used for the training, and only maybe hundreds of genes. And when the number of genes and number of cells goes smaller, we can see the difference between SL2SPA and other tools doesn't become that clear like here. And that explains that other tools can work on small data sets, but couldn't be scaled to larger data set. But SL2SPA can work on both low resolution and can work well on both low throughput and high throughput ST data. And after that, we want to see if SL2SPA have been overfitted. So we trained SL2SPA on one slide from static V2 data and predicted the location of voxels from another slide. So here is our training set, and here is our query, and here is our prediction. We can see we recovered the location of the cells very well. And we also investigated the expression of certain genes in our prediction. Like for this gene HPCA, we can see this gene is very highly expressed in the bottom right, and it goes lower when we move up or move left. And we also recovered the minor gene expression changes in our prediction. And similar results is also observed for another gene PCP4. We can see this gene is very highly expressed in the top right. When we move to the bottom left, the expression goes down. And we recover this minor gene expression change in our prediction as well. So the result shows that our SL2SPA can even recover minor gene expression change in our prediction. So after training on a lot of slides, now we try to predict the locations of single cells from single cell RNA-seq data. Here we trained our model on ST data and predicted the location of single cells. Here is the anatomic annotation for 4 cell types in mouse hippocampus. We can see that, and here we showed our predictions of single cells. We can see we predicted the location of those single cells very well. And we've also checked the expression of the genes in our prediction. The first column showed the true spatial gene expression of the 4 genes, and the second column showed our prediction. We can see we recovered the overall spatial gene expression pattern of the 4 genes in our prediction. So in our previous result, we trained on high resolution, near cellular resolution ST data. We also want to know if we can train our model on low resolution data like Bayesian data and predict the location of single cell RNA-seq data. And those results showed that we can predict the location of single cells to some extent, even when we train our model on Bayesian mouse brain data. Here is our result. So in the first row, we showed the marker genes of 3 cell types. In the second row, we showed our prediction of the 3 cell types. So here we trained on the slide a section from mouse 1, and we predicted the location of mouse 1. We can see the predicted location of the cell types match with the expression of the marker genes very well. And after that, we also want to know if we can do like cross-sample prediction. So we trained our model on section 1 and from mouse 1 and predicted the location of single cells from mouse 2. We can see we got similar result. That proves our model can also do cross-sample prediction for single cell location. And similarly, we've also investigated expression of 4 genes to see if we can predict the overall gene expression patterns. The first column showed the true expression pattern of the 4 genes, and the second column showed our prediction. We can see we trained on low resolution ST data, rhythm data, we can still recover the spatial gene expression pattern to some extent. So in the previous result, we predicting the location of single cells from SCRSec data. But we also want to know if we can expand our model to like more data from different protocols like SNRSec data. So we applied our model on Vism schizophrenia human upper cortex data and try to use it to predict the locations of single cells from SNRSec data. And here we showed the anatomic annotation for the ST data. So the layers from left to right, layer 1, 2, 3, and 5, 6. And here we showed our prediction. So we can say it's not as good as what we got from statistic V2 and Vism Mossburn data, but still we recovered the location of the single cells to some extent. And we can see more clearly, like in the box plot, we can see the single cells that are labeled with low layer are mainly predicted to the left side, and when the layer goes up, they are predicted to the right side. So we still recover the location of single cells from SNRSec data to some extent. And similarly, we showed investigated the expression of 4 genes to say if the match between the ST data and our prediction. And here we found 3 genes. So we can say it's not as good as what we got for the Vism data, but still we recovered the overall gene expression patterns in our prediction to some extent. And after predicting the location of single cells, we can further perform cell communication analysis based on our prediction. And you may want to know, why do we need to predict the location of single cells to do cell communication analysis? Why don't we just do cell communication analysis on ST data? That's because like in low resolution ST data or near resolution ST data, we don't know the cell types of a voxel because it's mixed, like there are many cells mixed in one voxel. So even if we do cell communication analysis, we don't know exactly which cell type is communicating with each cell type. So, but if we transform the ST data, low resolution ST data to single cell resolution and perform the cell communication analysis, we can know exactly which cell type is communicating with which cell type. That's why we need to predict the location of single cells. We have two strategies to converting the low resolution ST data into single cell resolution. For Vism and statistic, we can do random sampling. So for this, here we give an example. So we can randomly select one cell from the region between 5 micrometer to the center, and we select 4 cells between 5 micrometer and 50 micrometer. In this way, we constructed single cell resolution ST data based on the prediction of ST spot. And for statistic V2 data, we can do it more easily. We just selected the closest cell to the center bead. Because it's near single resolution, we only need to select the one cell for each bead. In this way, we can reconstruct a single cell resolution ST data. After reconstructing the single cell resolution ST data, we can apply cell communication inference tool to our data. Here, we applied Spartalk, published to a Spartalk to our reconstructed data, and we selected astrocyte and interneuron that communicates through HbGF and ERBB4. The reason we selected that because it has both high score and also have high number of ligand receptor co-expression number. So after finding the target interacting cell pairs, we can further visualize it in the predicted images. And specifically here, we are looking at paracrine signaling. So it's a way that a cell communicates with its nearby cells. And we can visualize this interaction in the predicted images very clearly. So here we can see exactly which cell is communicating with which cell. So the green spot is astrocyte, and the red spot is interneuron. If we zoom in, we can see exactly which cell is communicating with which cell. We can see it very clearly in our prediction. Also, we can also look into like which astrocyte is communicating with which interneurons through the specific ligand receptor HBE/GF and ERBB4. So here we don't show all astrocytes or interneurons that are communicating with each other. We only show them that communicate in using a certain ligand receptor. As we show here, we can see exactly which astrocyte is communicating with which interneuron using this ligand receptor. Yeah, and we can also do some downstream analysis. So here HBGF is the ligand, and after this ligand was sent to the receptor on the receiver cell, it will trigger many downstream analysis, and we can see it in this figure. So these are the genes influenced by the communication through the specific ligand receptor between the two cell types, and the blue boxes indicates those genes are impact factors. So with these genes, we can look into how the communication influence the functions of cells further. And we can also do some pathway analysis by running enrichment analysis for the genes that are influenced by the communication. We can know which pathways are influenced by this communication in this figure. And finally, I would like to mention that SS2Bar is now publicly available as a Python package, so you can install it in Python and run it very easily based on our tutorial. Now we only have one tutorial, but we will update more tutorial, so you can run our code by just following the tutorial. So just to summarize, so SS2Bar is an accurate tool to infer the spatial locations of single cells, and the prediction from SS2Bar can be used to study cell-cell communication further.